Hello everyone from Learning Academy Foundation. I'm Mr. Andrea Lombardo, the founder of this educational business, and today we are going to discuss about the structure of DNA. So, what is deoxyribose? What are nitrogen bonds and what are they used for? We will answer these questions in the next minutes of this video. Before talking about the actual structure of DNA, let's try to give a simple definition of this long polymer. DNA is the fundamental basis of life. We can imagine it as a long chain which is found inside every cell of the human body. Inside, we find the chromosomes, which contain all the genetic information that is transmitted from one individual to another. Each part of this chain is made up of simpler elements. From a chemical point of view, we can define DNA as an organic polymer made up of monomers called nucleotides, deoxyrribonucleotides. These nucleotides consist of three elements: a phosphate group, deoxyribose, that is the pentose sugar, and a nitrogenous base which binds to deoxyribose with a nitrogenous glycosidic bond. But now let's see something more about the structure of DNA and its function. DNA, like RNA, is a nucleic acid made up of subunits called nucleotides. Each nucleotide consists of three components: phosphate group, pentose sugar, and nitrogenous base. The reference sugar is deoxyribose, which can bind to four different nitrogenous bases: adenine, symbolized with AA, thymine, symbolized with T, guanine, symbolized with G, and cytosine, symbolized with C. The DNA molecule is made up of two paired polynucleotide chains wrapped around the same axis to form a double helix. Here are the main features of the same. These are complementary and anti-parallel chains. Adenine binds to thymine and vice versa, and guanine binds with cytosine and vice versa. The bonds between the nucleotides within each chain are covalent, while those that unite the two paired strands are hydrogen bonds. The helix has a constant diameter and right-handed winding. Where is DNA? DNA is found in the nucleus of all cells, of which carries the genetic code. The question is, what is the deoxyribose acid used for? Surely the most precious function ascribable to DNA is to contain the information necessary to make the body function. The wealth of data can be transmitted from one cell to another and from one organism to another. The molecule contains all the fundamental instructions for the synthesis of proteins that are important for building tissues and organs and for activating the biological and chemical processes that ensure the survival of the organism. Therefore, the most important function of DNA is to transmit hereditary characteristics from one individual to another. Now that we have seen the structure of DNA and its functions, let's see some historical references to its discovery, which represented a real evolution in the field of science. The first to isolate the DNA was the Swiss biochemist Friedrich Miescher, who in 1869 identified a microscopic substance contained in the pus of surgical bandage used. A few years later, in 1919, Phoebus Levin identified the nucleotide structure composed of nitrogen base, sugar, and phosphate. 1953 was an important year for DNA. It was in 1953, in fact, that Alfred Hershey and Martha Chase proved the role of DNA in heredity. Also the same year, thanks to the studies of Rosalind Franklin, English chemist physics, James Watson, and Francis Crick, the first model of DNA structure was presented. That is the double helix model as we know it today. I hope you have enjoyed the lesson. Thank you for your attention. Please remind, if you have not done yet, to subscribe and put like so that you can follow also the next contents of LAF. If you want to follow also from the social media, you find in the links in description about Facebook and Instagram social media. Thank you again. See you next time.